Hello, and welcome to this video presentation of chemistry system screening with Fusion QbD. First, let's briefly discuss what we mean by a chemistry system. The elements of a chemistry system include the stationary phase chemistry, the mobile phase chemistry, and in gradient methods, the slope, which represents a change in the chemistry conditions over time. These components actually represent a highly interactive chemistry system. Chemistry system screening means combining the major chemistry system effectors of peak retention, selectivity, and shape into a statistically rigorous designed experiment. With Fusion QbD, you can study any combination of LC parameters which can interactively affect method performance. In this chemistry system screening study example, we will combine multiple pH levels using the online preparation mode with multiple gradients covering a range of 10 to 25 minutes, in combination with multiple columns. Design of experiments at its heart is a model building methodology, which starts with the experimental design as an efficient statistical sampling of all possible combinations of the factors selected for study. In this illustration, combining pH and gradient time at 5 levels each, all possible combinations would yield 25 methods for study. Alternatively, the design of experiments sampling of the same experimental region would require 13 methods with 2 replications of the center point as an experimental error estimation data subset. The efficiency of design of experiments becomes even more apparent when you extend a sampling region across multiple combinations of a categorical variable like column type. In this case, all possible combinations would add up to 100 methods for 4 columns, whereas the design of experiments methodology would require 36 methods. The modeling aspect of the design of experiments methodology is to take the data that's generated from the statistical sampling and model it to fill in the information gaps to provide complete knowledge of the entire experimental region, thereby translating the region into a complete knowledge space. Let's now set up a chemistry system screening study within the Fusion QbD software program. We first select the instrument on which we will run the experiment. We then select the separation mode, reversed phase in this example. And lastly, we select the experiment type. In this case, we will use a chemistry system screening template, which is fully customizable. Making those selections brings us to the experiment setup window, which has been configured for our selected instrument. Our first setting is to define whether we are studying an isocratic method or a gradient method. Next, we select from among the available variables those we want included in experiment setup as either constants or study variables. The Activate Online Preparation panel is enabled and displayed when we have a quaternary pump. We'll discuss activated online preparation for pH in this example. With a quaternary pump system, we can use the Fusion QbD online preparation mode and assign acidic and basic forms of a buffer to different reservoirs. Fusion QbD will then proportionally blend the acidic and basic buffers to achieve our target experiment levels of pH. To set this up, we first click the buffer selector and then select a buffer from the drop-down list, which covers the pH region we want to study. We next select the specific target levels we want to include in the experiment. Note that if your preferred buffer system is not in the list, you can simply add your target pHs and buffer proportions of acid and base to the buffer selector setup grid, and Fusion QbD will blend the buffers to achieve your target levels of pH. We next select the strong solvent type. Here we're studying acetonitrile and assigned it to reservoir A. To add an additional strong solvent, such as methanol in a reversed phase method, we would change the number of strong solvents to 2, and this would enable us to add a second strong solvent and assign its reservoir. We next set up our slope variable for study. Fusion QbD enables you to set any pump program step as either a constant or a variable. Here we set the gradient time step as a variable with short and long gradient times to give us a broad range of slopes. To set up columns for study, we simply open the Number of Columns list box to identify the number of columns we're going to include in the study. We then enter the information on each column, and also, if a column switching valve is present, the valve position associated with the column. Fusion QbD automatically adds conditioning runs in the experiment whenever the chemistry system is changed between experiment runs. We can then also put in a conditioning time to make sure that we have sufficient column volumes of the mobile phase run through each column in anticipation of the next series of runs at the next chemistry. Once you've entered your experiment variable settings, clicking Update Setup Data will record those settings, and Fusion QbD will document them in a report. You then can click the Generate Design button. In the automated mode, Fusion QbD will automatically generate the most statistically defensible design for your study variables and bring you to the Design Reports window. In the Design Reports view, Fusion QbD presents an instrument report, which documents the exact system on which the experiment will be run, including the specific modules and their configurations, the experiment design, all experiment setup settings on which the design is based, and even the statistical information properties of the experiment design. On the Experiment Design Report, Fusion documents the required mobile phase compositions and their reservoir assignments, the column information, including the assigned valve positions and the names of the included columns, the experiment constant settings of all parameters which will be automatically set the same in all experiment run instrument methods, and the target level settings of all study parameters which will automatically be built into the instrument methods corresponding to the experiment runs in the CDS. Note that the Fusion QbD experiment design is structured for maximum data quality and minimum runtime. Fusion QbD maximizes the use of reservoirs and solvent selection valves, automates mobile phase preparation, incorporates column conditioning, and ramps on temperature and pH. Once we complete our review of the experiment design, clicking on the Export button will launch the Export Design Wizard, and Fusion QbD will export the design to your designated project within your CDS as a ready-to-run sequence with all required instrument methods and column conditioning methods pre-constructed for you. As we discussed, DOE is a model-building methodology, so let's briefly look at the different types of data we may want to model from a method development study. First, early chemistry screening or method scouting studies can yield very complex chromatograms, since the different experimental methods represent different mobile phase and stationary phase chemistry combinations. And in the early screening stage, we may not know how many individual compounds are in our sample, so accurately tracking peaks in these chromatograms is very problematic, especially considering the likelihood of significant coelution and changes in elution order between runs. This is why S-Matrix developed the trend responses. Trend responses are user-configurable metrics of chromatographic performance. Trend responses consist of an operator for the data query, a threshold acceptable performance value, and the user's desired response. Once you configure a trend response, Fusion QbD automatically extracts your user-defined trend response data from each experiment chromatogram. Trend responses require no peak tracking, just basic integration. Examples of trend responses include the number of integrated peaks, the most basic measure of selectivity of a given experimental method. You can configure a trend response to also determine the number of baseline resolved peaks in each chromatogram, the number of peaks with acceptable tailing, the number of peaks with acceptable signal-to-noise ratio, or any other chromatographic property of interest. Trend responses also automatically track peaks indirectly through their area. The max peak 1 operator represents the largest peak in the chromatogram, and here we've configured the max peak operator to report the resolution of the largest peak, and also the resolution of the immediately post-eluting peak to the largest peak. Trend responses also track and identify the first peak and last peak in each chromatogram, so any response data of interest can also be obtained automatically for those peaks. To import our trend responses for analysis, we access the Data Entry view, click the Import Responses button, and log into the chromatography data software. We then access the project containing our data, the results set associated with our experiment, and the derived 2D channel. Clicking the Next button enables us to access the Select Responses dialog, where we can set our trend responses. We will demonstrate some of the flexibility of the trend responses. First, we can remove a default trend response by simply unchecking the checkbox associated with the response. We can modify a performance threshold, for example, changing the tailing threshold from 1.2 to 1.5, and also adding a trend response. In this case, we will add the first peak trend response and the retention time result, since the first peak is a difficult to retain peak. Once our trend responses are defined, clicking the Next button will import all of the trend responses from the chromatograms automatically. We can then review and modify the trend responses as required for analysis. For example, clicking the Create Edit Response Data button launches the Create Edit Response Data dialog. We can then select the Max Peak 1 resolution data, and we observe that many of the runs have missing resolution results because the resolution was below the threshold for the chromatography data software to calculate a result. We can therefore add zeros to all of these values to better represent the lack of resolution of this critical peak pair. We can also modify the display precision associated with responses that will be used in graphs and reports. For example, the first peak retention time data is presented to 3 decimal places for display. We can modify the precision for display purposes by changing the number of decimal places from 3 to 2. We can also view the experiment chromatograms associated with our numerical trend response data by clicking the Show Chromatograms checkbox and selecting one or more runs to review. We can also sort the data entry grid, consolidating the grid by hiding the condition column runs, and also sorting on either a study variable or variables or a response. For example, selecting the column type variable groups the runs by column for quick review on a column-by-column basis. Once we have reviewed and updated our data as required, we go to the Data Analysis view, click the Analyze Data button, and in the automated mode, one click will analyze all the results and build all of the equations automatically. Once Fusion QbD completes the automated model building process, we can switch to the Best Overall Answer view and click Execute Search to find the best performing method overall. In the Search Setup Wizard, we define the specific performance goals required for the method according to our trend responses. We can leave the default goals of maximize for the Max Peak 1 resolution response and the first peak retention time response. But for the peak count-based trend responses, we would change the goal to target, since we know the exact number of peaks in our experiment sample. We then would put the specific goals for the number of peaks, the number of resolution results greater than or equal to 1.5, and also the goal for the number of peaks with tailing below 1.5. In the case of our target goals, we also define lower and upper bounds that define the threshold of what is an unacceptable result around our target value for the peak count responses. For the maximize response, we set an acceptability limit on the lower bound and also an upper bound that defines when we've met or exceeded expectations for the performance of the method. After these goals are set, clicking Finish initiates the search in which Fusion QbD will mathematically look for a method that meets all performance goals simultaneously. Once the search completes, a report is generated which identifies the method conditions yielding the best overall result. Once we view this report, we can switch to the acceptable performance region and create a graphical presentation of the best performing method and the region around the best method in which the chromatographic performance is consistent with our goals. In the main display, we have control over which variables are graphed and which constant settings of our non-graph variables to include. In the lower grid below the main graphical display, we have all of our settings for all of our response goals. To get oriented to this graph, we can eliminate most of the response settings and reduce our goal set to just the number of peaks and number of peaks which are baseline resolved. The upper graph reflects the fact that we've eliminated the additional response goals, and the unshaded region identifies the combinations of gradient time and pH which will yield all of our peaks baseline resolved. However, once we add in the requirements that they all be below 1.5 for tailing, we have fewer combinations of pH and gradient time, which will give us all of our peaks baseline resolved and with acceptable tailing. When we then include the minimum performance requirement for max peak 1 resolution, our API, and the minimum retention time for our first peak, we have a much reduced region of pH and gradient time combinations, which will simultaneously meet all goals for all these performance requirements. We can move the grid down to enlarge the graphed area to make it easier to read. And we can also change the designated column for the graph. Notice when we switch from the phenylhexyl column to the C18 column that there is no unshaded region. This means that there are no combinations of pH and gradient time within our study region that will meet all of our performance goals if we use the C18 column. The same is true of the shield column and also the T3 column. Only the phenylhexyl column gives us workable methods which will meet all of our performance goals simultaneously. Fusion QbD gives you complete control over the responses to include in a given graph and the performance goals you set for the responses. It also gives you complete control over the variables you graph and the ranges. Fusion QbD also lets you create multiple reports with different graph and numerical views of your experimental results. For example, this report displays a zoomed-in view of the workable region according to the current trend response goals and identifies the ranges of pH and gradient time, which can be promoted to an optimization experiment along with the CSH phenylhexyl column. That ends this video presentation of chemistry system screening with Fusion QbD. Make sure to view the follow-on presentation of method optimization with forced degradation study automation. Thank you for watching.